Hello to all again, and welcome to the Science of Childhood Cancer Lecture Series hosted by the St. Jude Comprehensive Cancer Center. Before I introduce Dr. Terry Fry, I would like to invite you to ask questions anytime during the talk. The best way is to use the chat to send the question to all panelists, or you can send it by email to the address on the screen. We will pause briefly about halfway through in case there are questions, and leave time for more before the end of the hour. Terry has also kindly offered to stay on for some time after the hour if anyone would like to stay and have further discussions. Before Terry gets started, let me introduce him. Terry is a professor of pediatrics, Robert and Kathleen Clark Endowed Chair, co-director of Cancer Immunology Initiative, and the director of Cancer Immunotherapy at the Children's Hospital Colorado and the University of Colorado School of Medicine. He was formerly the head of Hematologic Malignancies sections of the Pediatric Oncology branch at the NCI. Terry is among the first scientists to investigate CAR T-cell therapy and is a member of many medical societies, including American Cancer Society of Clinical Investigation. We are very pleased that Terry will give us a talk entitled The Future of CAR T-Cell for Pediatric Leukemia Beyond Antigen Targeting. Terry, thank you very much for sharing your latest research ideas. It's all yours now, Terry.
Ching Han, thank you very much for the kind introduction, and thank you all for attending. What I'm going to try to do today is, really talk a bit about, CAR T cells from the perspective of, our experience with a chimeric antigen receptor targeting an alternative antigen to CD19. That CD22, another B cell associated antigen. I think certainly the pediatric oncology community is learning how to use chimeric antigen receptor T cells targeting CD19 effectively in the context of pediatric leukemia, and certainly that's becoming a part of the standard paradigm of treatment. There's also been, I think, in the last 12 months to 2 years, some really elegant work starting to dive into the immune biology of T cells that incorporate chimeric antigen receptors, really trying to anticipate how best we might optimize these. What I'm going to do is really talk about, start with, our experience in a clinical trial, with what turned out to be an active CAR targeting a different antigen, and then really kind of go through how we took those observations and kind of went back into the laboratory to try to understand a bit what we were seeing in patients, and then try to think about how we could to use that information to modify in the context of the CD22 CAR. So, I don't have to remind the audience what we're talking about with chimeric antigen receptors, in the sense that these are genes that are introduced into T cells during an ex vivo manufacturing process, at least for the commercially available CARs. These cells are then expanded and then reinfused into patients, where they are quite effective at inducing remissions, as we know in pediatric leukemia. I'll point out that really, I think the intent of these receptors is to take advantage of advantageous physiological attributes of a T cell, and that is, the T cell's exquisite sensitivity for antigen, selectivity for antigen, the ability to expand in response to activation, the ability to traffic throughout the body, and what we're learning in leukemia, an important parameter, which is the ability to persist for at least some period of time. We all, I think, now know that across a number of early phase clinical trials, there was really a remarkable remission induction rate in CD19 positive leukemias. in children. This was data from our trial at the NCI, led by Trey Lee, with what ultimately became Yescarta. But this was the pediatric leukemia experience, about a 60 to 70% remission induction rate in that trial. And in some ways, what was remarkable was that across a number of early phase clinical trials conducted at a number of institutions, really there was very comparable efficacy in these early phase trials. What I think many people forget is that, there were a number of differences, in construct design, cell manufacturing platforms across these trials. And so in many ways, they weren't really identical drugs, so to speak, but nonetheless really induced, a comparable remission induction rate in patients that received, these genetically modified T cells in the early phase clinical trials. Everyone now is aware of the fact that, really in a remarkable event, the first FDA-approved indication for CAR T cells was actually a pediatric indication of pediatric leukemia, something we don't experience, unfortunately, very often in the pediatric oncology world. But this was then followed by the second approval, which was adult lymphoma. And this approval in the pediatric leukemia setting, which was, the construct of the drug that ultimately became Kymriah, was based on the licensing trial that was led by Shannon Mott and Steve Grupp, open at a number of sites around the country. And this was the data that was published in that original New England Journal of Medicine paper. So this is this curve shows duration of remission in patients who achieve remission treated with Kymriah on this licensing trial. And the thing that has definitely held up as we've gone into the real world experience is that certainly there's a very high remission induction rate, but we're also continuing to see issues with relapses that tend to occur in two phases. An early phase, which is attributed to lack of CAR T cell persistence. The duration of persistence that's needed is, I think being discussed, probably on the order of about 3 to 6 months, measured by often B cell aplasia. And then the second pattern of really more frustrating, in some ways, relapse associated with loss of CD19, a scenario where the CAR T cells do all they're supposed to do, but the leukemia becomes resistant. And so, around the time that the CD19 CAR trials, the early phase trials, were being conducted, Rima Sorrentis, Walid Hasso, a number of other folks at the NCI began working on a CD22 CAR, and it was really based on a program in CD22 targeted immunotherapy that was led by Ira Paston, really in hairy cell leukemia. And the lead construct that they ultimately took in the clinic and ultimately became approved for hairy cell leukemia was a toxin conjugate that was based on a CD22 binder known as BL22 that was eventually affinity enhanced to generate what was referred to as HA22, high affinity 22. Again, very effective in hairy cell leukemia as a toxin conjugate. But when Rimis and Walid took the logical step and took this high affinity binder and put it into the preclinical model systems that we were using at the time, and many people are using now, this particular CAR that incorporated HA22, this high affinity binder, was completely inactive. And I give them credit because rather than giving up, they went back to the freezer, so to speak, and pulled out a series of other binders, including M971, which was really discarded by Ira and Mitko Dimitrov, who was the person who created these binders from his phage displays. They're fully human binders. And it was really discarded because it was relatively low affinity for CD22. But it turned out to be very effective as a chimeric antigen receptor in these early phase clinical trials, as you can see from this mouse data below, with clearance of tumors. And, to some extent, the community already knew potentially why that was, because Ollie Press in Seattle had already shown that that membrane proximal binding seemed to be important for CD22 CAR efficacy in a paper that was actually published in 2008. And so that turned out to be, so to speak, the magic with M971. And I think that's continued to be true as people have developed alternative CD22 targeted constructs. So in my lab, we basically just converted this from a CD28 endodomain containing CAR to one that contained 4-1BB. This is the endodomain that's contained in Kymriah and seems to mediate longer persistence of CAR T cells. We were already appreciating that as an important parameter, and so modified the original CD28 version to the 4-1BB endodomain. So this clinical trial, which was probably the largest phase 1 clinical trial I've ever been involved in, opened in 2015. It's actually still open. Nirali Shah is really leading these efforts right now. There was an original publication in 2017 that reported on the first 40 patients or so. Follow-up publication this year reporting on now up to about 60 patients. I think Nirali's up close to 80. The remission induction rate is held up at about 70 to 80%. Importantly, this has not fallen off, as we've seen the majority of the patients enrolled in the study more recently be CD19 negative relapses after CD19 targeted CAR T cell therapy. The other thing that we learned from this trial was that modifications to the manufacturing process can have an impact on potency and toxicity. This CAR has had, in addition to a pretty high, to a very high efficacy in CD19 resistant, CD19 CAR resistant leukemias, has resulted in relatively low neurotoxicity, but interestingly has had a slightly different, has had an increase in a toxicity that is often referred to as HLH-like toxicity, in the sense that it sort of mimics hemophagocytic lymphohistiocytosis, but isn't exactly the same. It occurs with CD19 CAR T cells, but we've seen it more frequently with the CD22 CAR. So this trial is currently, again, still open. The CAR itself received breakthrough designation by the FDA, and Nirali and team are actively working on developing the licensing trial for this particular CAR. But, one of the things I want to kind of go back to is that pretty early on in the CD22 CAR development work, we were already starting to appreciate the fact that the CD22 CAR was a bit different than the CD19 CARs that we were working with at the time at the NCI. And I'll point out that really one of the earliest publications that really alerted us to the fact that not all CARs are created equal was this paper by Adrian Long and Crystal Mako's lab, where she kind of looked at the characteristics of a CAR targeting GD2 that many of you are familiar with. Has an antigen that's expressed on a number of solid tumors, including neuroblastoma. And one of the things that Adrian noted was that CARs that express this GD2, T cells that express this GD2 CAR were prone to exhaustion, and this occurred even in the absence of antigen. And what she showed very elegantly in this paper was that what happened with the GD2 CAR is that it tended to cluster, as shown in that imaging figure C that was in the paper, that there was this clustering of these CAR receptors on the surface in the absence of antigen, and this was in contrast to CD19 CARs containing FMC63, which distributed fairly uniformly on the membrane. And what she showed in this paper was that this led to chronic signaling and exhaustion of these cells. What I'll point out is that in the supplemental data in this particular paper was data on the M971 CAR. And what Adrian showed was that this particular CAR, the CD22 CAR, had sort of an intermediate phenotype, in the sense that it didn't exhaust quite as much as the GD2 CAR T cells, but it certainly exhausted more than the CD19 CAR T cells. And Kelsey Wanhanen and Hein Kin in my lab did some imaging actually looking at the CD22 CAR, and like the GD2 CAR, there was some clustering on the surface that was antigen independent. And so at that point already, we knew that there were some differences with this CD22 CAR from the CD19 CAR. So, so the other point I want to make here is that certainly since this early publication from Adrian and Crystal's group there's been a number of publications that have really identified that that the structure of the CAR really does have an impact on how these cells behave when ultimately introduced in vivo, and really how they behave in vitro model systems. And so this is a schematic of a CAR. So you basically have a binding domain typically derived from an antibody, which is a heavy and light chain separated by a linker, and I'll come back to that in a bit. A hinge region that attaches this binding domain to the endodomain, a transmembrane region, CD3 zeta chain, and in the so-called second generation CARs, the ones that are active in the clinic, a co-stimulatory domain. And the two co-stimulatory domains that are most commonly used are CD28 and 41BB, as I've already mentioned, although other alternative domains are being used. And I would say that I don't have time to summarize sort of what's really a growing body of literature that suggests that all of these components of the CAR structure itself can have an impact on how these CARs perform. And so really what you have is a very complex drug that performs depending on how it is built. So I want to go back for a moment to the CD22 CAR. And you know, one of the quirks of this CAR was that when Mitko made this binder, he essentially created this single chain fragment variable region with a very short linker, a single G4S linker. Most of the CARs in the clinic actually have at least 15 to 18 amino acids between the heavy and light chain. And the thought is that that structure allows for the heavy and light chain to associate in the same receptor. You know, I think the prediction would be that with a single G4S, that the ability for this for these heavy light chains to connect is going to be limited. And I will tell you that the group from Penn, Marco Ruella, Sarah Gill, and more recently Nathan Singh, who is now an independent investigator at Washington University in St. Louis, really did some very elegant work with a CD22 CAR that they introduced into the clinic that was essentially the M971 binding domain, but instead of using a short linker, the typical G4S linker that contains 15 amino acids between the heavy and light chain. And it turned out that that particular CAR, referred to on the right there as the CD22 long CAR, had activity in the preclinical models. But when they introduced it into patients and opened their clinical trial, it was certainly less active than the short linker CD22 CAR. So somewhat surprisingly, the short linkers seemed to be important for efficacy in humans, although I will tell you that it did show activity in the preclinical studies. You know, Nathan has really gone on to do some elegant work and has shown that essentially by lengthening the linker, what happens is that you convert this sort of clustering propensity of the CD22 CAR into now a CAR that distributes uniformly on the surface. So it looks pretty in the images, but again, turns out not to be as effective at eradicating leukemia in patients. So, you know, I want to go back now and I want to just talk about another observation from the clinical trial, because not only is the CD22 CAR somewhat different than the CD19 CAR in terms of what seems to be the optimal format, but the antigen itself is quite different from CD19. And it's different in a number of ways. One, it tends to be expressed at lower levels on the surface of on leukemia, at least on average, compared to CD19. The number of CD19 molecules per typical ALL blast is about 10 to 15,000, whereas for CD22 it tends to be about 3 to 5,000. Lower in some patients, like those with KMT2A rearrangements. The second thing that we noted was that in the context of the clinical trial was that patients would achieve remission, but they tended to relapse with leukemia that actually had lower expression of CD22, not loss of CD22, but just a decrease to the point that the CAR was seemed to be no longer able to identify the leukemic cells, and that is shown in this data here. So one of the first things that we just wanted to ask was whether or not this observation that patients were relapsing with a change in CD22 expression levels was association, or whether there really was a mechanistic connection between those two observations. And so to do that, Sneha Ramakrishna and others in the lab generated a series of cell lines based on NALM6, a B-cell ALL cell line that is commonly used for these sorts of studies. Again, the parental version of NALM6 tends to express about 2,000 CD22 molecules per cell. So Sneha and team CRISPR'd out CD22 and then reintroduced CD22 at different expression levels, generating this low cell line, and this is at about 500 to 1,000 molecules per cell, which is sort of typical for the site density of the relapses we were seeing in the context of the clinical trial. And then this other cell line that had very high expression levels, even beyond that seen with CD19. And as shown on the right there, indeed, what Sneha found was that when you looked at the activity of the M971, the clinically active CAR, against the low site density expressing leukemia, this blue shown here, what she saw was that although there was some activity at higher cell doses, that's shown in the purple line, she was never able to get full eradication of the low site density leukemia with the CD22 CAR. And in vivo, this was associated with less expansion of the CD22 CAR T cells in the context of low antigen expressing leukemias. So Zach Walsh in my lab in Colorado really took a deeper dive into what happened to the T cells expressing a CD22 CAR when they were activated by leukemia cells expressing different levels of antigen. And what Zach found was that indeed there was sort of a titration of different downstream effects. CD107A is expressed on the surface of cells that undergo degranulation. And so in essence is an indirect measurement of cytotoxicity. CD69 and CD25 are both activation markers, and Zach has done a whole series of downstream assays looking at the functionality of T cells in the context of low versus high site density expressing leukemias. And I can tell you that although they all titrate up with antigen, the point at which they plateau varies depending on the downstream effect that you look at. And so, for example, if you look at CD107A, it really sort of peaks at the parental NALM6 cell line. When you look at some of the activation markers, it continues to go up as you increase antigen density, even to those extremely high levels found in that cell line on the far right. So one of the next obvious questions was to sort of think through how to deal with the fact that low antigen expression seemed to be a liability with the CD22 CAR. And so Mitko and his team really generated a series of binders, all based on M971, but with higher affinity for the antigen. And importantly, these modifications to the M971 construct were really 2 to 3 amino acids. And although we didn't do formal epitope mapping, the prediction would be that they all bound to the same epitope on the CD22 molecule. And ultimately, what we came up with as the binder that seemed to make the most sense to test further was this version 1 of M971. And those are the association and dissociation curves on the left there. I'll point out that the scale is different between the standard M971 and the high affinity version. So this is the on rate for the high affinity version. The scale goes up to 120 in terms of response, RUs, and then whereas this goes up to 35. So much higher affinity, also at a slower off rate. We then took this, and one thing I want to come back to later is that the binder that Mika ultimately made turns out to be important. We'll come back to this later. Was high affinity from the standpoint of the heavy and light chain, but this was actually connected by a longer linker. And so the initial series of experiments we conducted were with this version of M971. And what we found was that indeed, when you looked at granzyme B, in response to CD22 expressing leukemias, it did seem to show more granzyme B production, particularly at low E to T ratios, which we think is important. And we can come back to that in a little bit more. But perhaps even more importantly, one of the things that we noted was that this high affinity CAR appeared to demonstrate enhanced efficacy in the cell line against the NALM6 cell lines, in particular, the cell line that expressed lower levels of CD22. So it did appear, at least in the context of CD22, that enhancing the affinity of the binder seemed to improve efficacy in vivo against low antigen expressing leukemias. And this was associated with improved expansion at day 21. One of the liabilities we had noted with CD22 CARs expanding in the context of low antigen expression. So Cole DeGollier, in my lab, a graduate student in my lab at the University of Colorado, has really started to take a much more careful look at the impact of site density on T cell behavior. And one of the things that Cole has really noted is that site density and affinity not only affect T cell response, in a mouse or in a dish, so to speak, looking at the global response of all of the responding T cells, but really seem to affect the response at the single cell level. And this is just looking at intracellular cytokine staining, looking at the percentage of cells in a dish that are able to secrete IL-2, interferon gamma, or both in response to CD22 expressing leukemias. And what you see is that this affinity enhancement and site density both have an impact on the percentage of cells responding. And certainly Cole, with his work, is intending to take a much more careful look at that. So we had the brilliant idea of sort of saying, look, high affinity, this short linker seems to be important for the CD22 CAR based on our experience in the clinical trial, and then the pen experience in the clinical trial. And then we had this high affinity binder that seemed to improve efficacy in the context of low site density expressing leukemias. So we decided to shorten the linker length in the high affinity CAR. And what I will tell you is that we did not end up with an additive benefit in the context of responses to leukemia. This is looking at imaging in mice that were injected or engrafted with low antigen expressing NALM6. And what you see is that certainly the high affinity version of the, this is parental NALM6 I'm showing here, that the high affinity version with the short linker actually essentially killed the activity of the CAR, as shown here in the red, resulted in a non-curative effect. And interestingly, was associated with, so CD39 is a marker that Crystal Makel's lab has sort of identified as important in CAR T cell functionality, sort of a quasi-exhaustion marker. Whereas the long linker version of that high affinity CAR seemed to be less exhausted, when you then introduce the short linker version of that, you now got expression of CD39 on these CAR T cells. So I'm going to pause for a moment. I don't see any questions on the chat. I'm certainly happy to take questions at this moment if anybody has any from what I've said so far. And I'll continue to pay attention to that. But what I'd like to do now in the last sort of 20 minutes or so before we get to questions at the end is to really highlight some work that Eric Kohler, who's a faculty member at the University of Colorado, started with me at the NCI and has really developed his own research program here. And Eric really very early on began thinking about what I think is a critical question, which is when you introduce these chimeric receptors into a T cell, are we really simply redirecting their specificity for a different antigen? Or are we in effect really reprogramming these T cells to do things that are slightly different than a physiologic T cell might do when activated through the T cell receptor? And so to do this, Eric took advantage of a mouse model that we had in the lab, which was based on a pre-B cell leukemia that was a syngeneic pre-B cell leukemia on a C57 Black 6 background, generated in mice that expressed TCF3 translocated human translocation with PBX1, resulting in this immature B cell malignancy. It engrafts in mice at very low cell doses. And importantly, using a CAR that was developed, a syngeneic mouse CAR developed by Jim Kokendorfer, we're able to show that we could make mouse CAR T cells, introduce these into mice with this syngeneic leukemia, and induce long-term remissions in these mice. Importantly, this required lymphodepletion. I think something that we've all come to appreciate in the field. So what Eric did was to really take advantage of this model as well as sort of immune biology systems, including T cell receptor transgenic T cells, in which the T cells have fixed specificity for known antigen. And the system that Eric worked with was the OT1 system to begin with. And these are T cells that have a T cell receptor that recognizes, they're CD8 positive T cells. They recognize an antigen derived from ovalbumin. And then Eric introduced our CD19 chimeric antigen receptor into these T cells. So now you had a T cell with known TCR specificity and known CAR specificity. He then generated a series of cell lines based on our syngeneic mouse leukemia that expressed CD19, the wild type version, CD19 and ovalbumin, which is the wild type version into which Eric introduced the ovalbumin protein, or an ovalbumin positive CD19 negative in which the CD19 was CRISPR'd out. And then did a series of experiments using this cell-based stimulation system to really interrogate the biology of these T cells. And so as you predict that basically you get no signaling with the double negatives, you get CAR signaling with the 19 positives, you get TCR signaling with the OVA positives, and then you would signal through both receptors with a cell line that had 19 and ovalbumin. And he used this then to go back and ask questions about the behavior of these cells downstream of these two different receptors. And the first question that Eric addressed was whether these behaved as they should. And this is again CD107A, looking at degranulation. I won't go into this in great detail, but using this assay and a number of other assays, Eric was able to show that both receptors were active on the surface of these T cells. And interestingly, when he did a very simple initial experiment and just looked at the ability for T cells with the ovalbumin, the OT1 TCR and the CD19 CAR, the ability for these cells to eradicate leukemia in a dish. This is an incucyte assay. In fact, the CAR tended to be more potent at eradicating leukemia than the T cell receptor. And the next question that Eric asked was whether or not which receptor was most efficient at clearing leukemia in a mouse. And interestingly, in contrast to what he observed in the in vitro setting, the IncuCyte assay, Eric actually saw that the T cell receptor was actually more efficient at eradicating leukemia in a mouse. And this seemed to be most apparent when he administered low doses of CAR-OT1 cells, so-called stress dosing of this therapeutic cell. Interestingly, in the high dose situation, Eric saw that at early time points, both the CAR and the TCR were able to eradicate the leukemia, at least leukemia that was identifiable by flow cytometry. And so really what was happening in the mice shown in this blue line, this is really a relapse scenario where there was early eradication, but then relapse after, when the CAR receptor was engaged, whereas the TCR was able to eradicate leukemia long term. So, really the observations then from this work was that the inability of a CAR to fully recapitulate the T cell receptor results in differences in both functional outcomes and, this is a mouse study, but really, quote, clinical outcomes, that is eradication of leukemia in an in vivo model system. So Eric has really gone on to do what I think is some very elegant work, trying to then ask questions about the phosphoproteomic profile of cells that are stimulated through either the chimeric antigen receptor or the T cell receptor using the system. And the approach he took was sort of an agnostic look at phosphoproteomics in a way, using SILAC, in which T cells and target cells are labeled with heavy and light peptides. There's stimulation, and then there's mass spec. Using mass spec, he's able to ask about global phosphoproteomic profiles that are downstream of TCR activation, CAR activation, or both. And what Eric has really found is that there are a series of proteins that are differentially phosphorylated downstream of the chimeric antigen receptor versus the T cell receptor. And really one of the biggest differences is really those proteins that are dephosphorylated in the context of CAR stimulation. And I'll come back to that in a moment. So Eric then took this data and then took a more targeted phosphoproteomic approach to really understand what was happening in some of these pathways in cells stimulated to the CAR or the TCR. What he found was that CAR stimulation tended to lead to sustained proximal signaling, and this resulted in enhanced activation of proteins in the NF-kappa B pathway relative to TCR stimulation. And this is, so the proximal is the PZAP70 shown here, and then NF-kappa B and PLC gamma. Again, shown to be more highly phosphorylated, if you will, downstream of the CAR versus the TCR. But this was not generalizable, because when he looked at MAP kinase pathways, what Eric noted was that, in fact, PERK, which is sort of the funnel downstream of the MAP kinase pathway in a T cell, tended to be less efficiently phosphorylated in the context of CAR stimulation versus TCR stimulation. He then went on to sort of show that this enhanced activation of ERK by the TCR tends to correlate with some predicted in vivo behavior. Importantly, because this is a syngeneic model, Eric was able to then take these cells, put them back into mice, and ask questions about how they behave in a fully immune competent host. And what Eric has found is that cells stimulated via the TCR tend to expand better, but most importantly, perhaps, they tend to persist better, particularly in the absence of antigen. And this is a manuscript that's in preparation, going back for resubmission. But really, I think importantly shows that the programming of the cell really does indeed seem to be different when it's stimulated through a CAR via, as opposed to a T cell receptor. And so to summarize Eric's work, I think he very elegantly showed that some of the structural and physical differences of CARs relative to T cell receptors results in really a unique signaling path profile. And this tends to correlate with some of the altered in vivo biology and potency. And sort of just at a very high level, I think what Eric's sort of model suggests is that In a T cell receptor, you've got more efficient phosphorylation of ERK that results in anti-apoptotic pathways, increased IL-2, better persistence. Whereas in the CAR, with this sort of skewing towards NF-kappa B, you tend to get activation, proliferation, target cell lysis. You get very efficient initial clearance of leukemia, but this more antigen-dependent persistence phenomenon that he tends to see with the chimeric antigen receptor. And I think all of this is really ultimately going to be critical as we think about the desired characteristics of this therapy when we introduce it into patients. So I want to go back for a moment then and sort of take Eric's observations regarding signaling in the context of the syngeneic model system and sort of show how Eric has applied this then to the CD22 CAR. So we went back and he looked at the site density model and asked questions now, not just about cytokine production and in vivo activity and in vitro activity, but really asked about the phosphoproteomics in the context of low antigen expression. And indeed, what Eric saw was that, that p-ERK or less efficient phosphorylation of p-ERK tended to correlate with lower expression of CD22 on the surface of the target cell, and that's shown here. So in some ways, really, since we didn't have, the ability to really compare CAR to TCR, this then becomes sort of a way of sort of measuring some of the differences. And then looking a bit more carefully at signaling pathways, what Eric noted was that in the context of low antigen, you tend to uncover this less efficient phosphorylation of ERK, and as would be predicted from the mouse studies, the NF-kappa B pathway actually tends to hold up better even in the context of low antigen expressing leukemias. So the other thing that sort of emerged was, as I showed you, the affinity enhancements to the CD22 CAR resulted in improved activity in the preclinical models. And indeed, the other thing that Eric has noted is that this affinity enhancement also results in a greater recruitment of T cells that are able to phosphorylate ERK in a dish in response to particularly to low site density expressing leukemias, as shown here in this particular figure. So before I go on just to the last 5 minutes or so, there were I see a couple of questions that have come up, and I'll just go ahead and try to answer these. So one of the questions was, in a post-transplant extramedullary relapse with 100% donor cells, is it possible to use CAR T cells? There is no IHC marker for 19 or 22 on the tissue to guide therapy. I guess I'll answer that just very quickly and say that I do think it's important to look at antigen expression, since we certainly know that it can change pretty effectively in the context of CD19 targeted immunotherapy. There's a question about manufacturing differences that I mentioned in the context of the 22 CAR trial. If I may, I think I'd like to get back to that one at the end, because I think that's a really critical question, and I'd like to maybe address that at the end. Perhaps one that's more relevant to the section that I just completed. There was a question about whether the p-ERK based on the p-ERK requirement for T cells to be activated in clear leukemia cells, do you think that MEK inhibitor-based therapies would impair immunotherapy and explain the shortcomings of such therapies? It's a really interesting question, and I will tell you that we've begun to look at MEK inhibition as a potential way to improve functionality of CAR T cells in response to low site density antigens, but I really do think that's an important question, and unfortunately I don't have really the data to answer that, but I think it's an important point. So I just want to go back then and take the last few minutes and just finish up the last part of this talk. So I've shown you a lot of data that relates to a very specific CAR targeting CD22, and hopefully convinced you that our experience with the CD22 CAR suggests that it's going to be a more challenging antigen, and One where I think even slight modifications to the design of the CAR can have an impact, a significant impact on the potency of the cells, not only in preclinical studies, but even in patients. And I think this is an important lesson for all of us that are sort of developing CARs targeting novel antigens. And so I want to just take a step back then and ask, so what might be the relevance of the data I've shown you in the context of other CAR T cells? So, my lab, quite a while ago, became interested in sort of thinking about, and many others obviously have been interested in thinking about how to extend, CAR T cell therapy to AML. A logical place to start was with CD33, a validated antigen in the context of AML. And in my lab, and then in collaboration with Sarah Tation, really spent quite a bit of time trying to do some preclinical optimization work, really built around binders. We used two binders that had been in the clinic, two antibodies, lintuzumab and gemtuzumab. And then looked at activity, depending on the co-stimulatory domain that was used, and came up with a lintuzumab CAR with a CD28 co-stimulatory domain, appearing to be the optimal construct for what ultimately, I'm going to show you in a moment, became a clinical trial that is now open at the NCI and CHOP, hopefully opening soon at other sites. But I'll point out that it turns out that CD33 is in the same family of molecules as CD22, in that it is referred to as a cyclic. And like CD22, it tends to be expressed at relatively low densities on the surface of AML, more comparable to that seen with CD22, certainly than with CD19. And it also tends to, we have seen in the PDX models, sort of modulate on the surface, or sort of change on the surface of the cell, sort of go up and down with therapy. And so it very well may be the case that we're going to encounter some of the same things that we've seen with CD22 CAR in the context of the CD33 CAR. Just as a reminder, this, as I mentioned, this CAR is now in the clinic as a first-in-child trial. The first two patients have been enrolled and infused. That's a standard phase 1 dose escalation study. This trial is being coordinated through the CIBMTR with funding from the Pediatric Blood and Marrow Transplant Consortium through a St. Baldrick's grant. We're looking forward to seeing how this performs. We're only at dose level 1, and hopefully we'll be able to escalate to more active doses. We have seen some CRS in some of the earliest patients. So the other place where I think the work that I've shown you becomes relevant is in this sort of concept of targeting more than one antigen. So with the loss of CD19, as occurring in 30 to 50% of patients with leukemia, with the activity of the CD22 CAR, introduces the possibility of incorporating both of these therapies up front, perhaps to try to reduce the incidence of antigen loss as a form of relapse. And so my lab, began working with a number of different ways to do this. A bivalent that is currently in clinical trials at Stanford, a single construct with two binding domains. But perhaps the way that we have favored is using a bicistronic vector to drive expression of two full-length CARs on the surface of the same T cell. And Haim Kin in my lab at the NCI went through a bunch of different iterations, came up with a construct which is the CD22 with 41BB, the CD19 with CD28. This construct, the clinical grade vector, has been manufactured, and this will be entering clinical trials at the University of Colorado and NCI, hopefully within the next year. But, and this is just showing the preclinical data showing that this CAR is effective at eradicating both CD19 positive and CD19 negative, CD22, leukemias in our preclinical model systems. But the thing that I think is really interesting about this is that not only do you put two antigen specificities on the surface of the cell, but you have now two endodomains, two different co-stimulatory domains. And really what you end up with is, at least in the context of cells expressing both antigens, is really a signal integration of those two signals. And I can tell you that when you look carefully at gene expression data, metabolomics, et cetera, you really get very interesting features of the T cells that that aren't necessarily predicted based on what you see with single CAR expressing T cells. And so I think as we get into these more complex formats, I think these signaling characteristics, et cetera, are going to become even more important. So, my summary slide before we jump to questions, I think it's pretty clear that single antigens comparable to 19, I think are going to be difficult, maybe impossible to find. We certainly know that antigen modulation and even more complex patterns, these lineage switch phenomenons that we're seeing in the KMT2A rearranged leukemias are going to emerge, I think, pretty frequently. This is happening even in myeloma with BCMA. I think it's more common with lymphoma than we appreciate because often we don't look since it requires a biopsy at relapse. I hopefully convinced you that CAR formats, at least the current formats that we're using, do not fully recapitulate T cell biology. That can be a positive or a negative depending what you're trying to achieve. But I think most importantly, I think optimal construct design is really going to depend on the desired in vivo behavior that you want to get out of your T cell. And this has to be considered in the context of the antigen or the specific tumor that one is attempting to target. And so with that, I certainly want to thank, I thank most of the people who did the work on the individual slides. Really a terrific team in the pediatric oncology branch that, led to the clinical trial. My lab, my new lab in Colorado, and all of the supporting folks in my new lab in Colorado. Really want to highlight Eric and his newly forming lab at the University of Colorado and funding sources, and certainly patients and their families for agreeing to participate in these trials. So with that, I'll stop. And before I start taking questions, there's a few additional ones that popped up on the chat. Let me just make sure. Let me come back to that question about manufacturing for a moment. I will tell you that so it's in the JCO paper. Remarkably, the manufacturing modification that we made in the middle of the CD22 CAR was really simply doing a T cell selection of the apheresis product. We noted early on that the messiness of the apheresis products, which contain monocytes, NK cells, et cetera, really resulted in, some cases, inability to manufacture a CAR T cell product. Certainly some differences in how these cells expanded and transduced in vitro. And so we just added in a T cell selection, a seemingly minor modification, ended up reopening the trial, started seeing a very high incidence of that HLH-like toxicity that I mentioned, and then had to actually dose de-escalate by a half a log, and that is the dose that we're currently using in the clinical trial. So really remarkably minor modification, yet certainly some differences in how these cells behave. The other question that showed up in the chat box, okay, I'm reading these for the first time. So when comparing short versus long linkers in the scFVs, did you look at whether the short linker causes pairs of receptors, this dimerization? That might explain the clustering. So I absolutely agree. And in fact, really credit to Nathan Singh and his work. I think this is really the model that he's looking at. And so I believe what really we're doing with the short linker is forcing some dimerization of these receptors. This results in some tonic signaling, and in a way primes the T cell to respond to low density antigen. So those of you on the phone or online who might be immunologists and can know some history of immunology, Ron Germain's lab many years ago showed that one of the features of a T cell in the periphery is that it gets sort of a low level, quote, tickling through the T cell receptor by cross-reactive self-antigens. This results in some proximal phosphorylation, that sort of basal phosphorylation, and this seems to be important in the T cell response. And so, I really am beginning to think that what's happening with a chimeric antigen receptor, particularly in the context of low antigen density, is a similar phenomenon. So the other question that came up was, have you looked at other targets SCFEs for the long linker high affinity SCFE relationship? Is this going to be target specific and should be empirically tested? So what I will tell you is that there's published data that, at least in the context of CD19, that actually dropping the affinity of the binder seems to be a good thing. And you know, in some ways that would make some sense based on what I mentioned earlier about the expression level of CD19. I almost think that, you know, really that in a 19 CAR, maybe you don't want that affinity. Whereas with 22, you probably, you know, it seems like you need a higher affinity, or that can be a benefit. So I completely agree. I think that what you want in terms of affinity, what that sweet spot might be, really is going to depend on the antigen you're going after, and not just sort of the protein itself, but really the expression level of that antigen on the surface of the target cells. Another question that came up is, can you comment on about CD22 with drugs? So, you know, in that paper that I mentioned, thank you for the question, by Sneha Ramakrishna, where she made the observation about low antigen density and CD22 CAR activation in the T cell. The other really important piece of that paper, and maybe the primary piece of that paper, was that she was able to kind of repurpose an old drug called briastatin. This is a PKC inhibitor. Actually, it tends to be a broad PKC inhibitor, broader than some. It was actually tested as a cancer therapeutic, and in the early phase studies with dose escalation resulted in some toxicity. Turns out that briastatin upregulates CD22 antigen on the surface of target cells, really an observation Ira Paston made initially. And one of the things that, so Sneha in that paper was able to show that a combination of briastatin plus CD22 CAR resulted in enhanced efficacy in the preclinical models. And so the NCI is rapidly gearing up to do a briastatin co-treatment study, really to try to look at that effect. It turns out that you can upregulate CD22 at relatively low concentration, so you might be able to avoid those toxic effects. The other question, which is an important one, and I know we're right at 12, but I'll answer it very quickly, is what are your thoughts on what the best management is of ALL patients who achieve a CR post-CAR, proceed or go to transplant? And boy, I'm going to answer that by saying that I don't know. And honestly, I think there are some characteristics that we can use in some parameters to follow in the patient, NGS sequencing, you know, for very deep remissions and persistence of the CAR. But ultimately, I end up in many of these situations presenting both options to the families and having a conversation, because I'm not sure I know the right answer. I certainly think having effective salvage is going to be important. So with that, I think we're right at 12 o'clock. I'm going to stop. I think that turn it over.
Thanks, Terry, for excellent lecture and outstanding answers. I just want to remind everyone that Terry has kindly offered to stay on after the hour if anyone has further questions or discussions. Let me ask you one. So in the dual CD19, CD22 CAR T cells, can you comment on the differences in response between the single bispecific CAR and one using two separate CARs and dual transduction?
Yeah, yeah, thank you, Ching-Han. It's an important point. So Stanford has certainly presented their data, and the CD19 by CD22, the bivalent that we made, you know, has activity in lymphoma and leukemia. You know, one of the things that we've always been concerned about is maybe not surprising based on our experience with the CD22 CAR, is it was very difficult to retain potency of the CD22 portion of that bivalent, you know, when we made those constructs. And so in the paper in molecular therapy oncolytics that Hein Kin published, we published a number of years ago, she described sort of the iterative development of that bivalent. And boy, it was not easy to even get activity of the CD22 portion through the generation of that bivalent. But even the best one we came up with, it seemed like when we look carefully at potency assays, et cetera, that we lost some of the potency of the CD22 with that bivalent. And, we'll see what the Stanford data shows. So, for practical reasons, we ended up emerging at the bicistronic. And again, I think, there's an opportunity to potentially mediate some integration of those two signals in a way that might generate some desirable characteristics in the T cell. So I'll be interested to see how that plays out. With other antigens, there's other bivalent single constructs with two binders that are in the clinic, CD19, CD20, as well as a number of other bivalents that are in the clinic. And certainly it seems like in some of those scenarios, it might have been a little easier to make the bivalents. But for 1922, I tend to favor the bicistronic.
So how about using two separation populations, CD19, one population, CD22?
Yep. So I guess that's a co-infusion, and certainly, makes a lot of sense and might reduce the complexity. Some of the early experience, not published, but certainly presented in abstract form from Seattle, Rebecca Gardner, Mike Jensen, the group in Seattle. They opened a clinical trial doing a co-infusion of 19 CAR T cells and 22 CAR T cells. And at least anecdotally, what seems to have happened is that the CD19 CAR T cells tend to, for lack of a better term, outcompete the CD22 CAR T cells. So, I think they ended up closing that trial. Now, I will tell you, I believe they made slight modifications to their CD22 CAR. And as I hopefully convinced you today, modifications to that CAR can lead to unexpected changes in efficacy. So, I think the answer to that question may not be completely answered yet, at least in humans. But we do have some preclinical studies where we did co-infusion experiments in mice, and I will tell you that some of this is published in that molecular therapy paper that I mentioned that Hang and I published a couple years ago. We saw the same phenomenon where you didn't always get, both of the populations of T cells to persist to the extent that you wanted them to.
So does the type of T cells used to make CAR T cell make a difference in the treatment response?
I'm going to answer that very simply and say yes. I think that certainly it's pretty clear to me, and I think most in the field, that the quality of the T cells that you start with has an impact on the response. And interestingly, when you think about what I showed you, anyone who makes CAR T cells and tests them in human makes CARs and tests them in human T cells, knows that there's donor to donor variability in transduction and expansion, potency. And these are healthy donors. These are donors without leukemia or lymphoma. So we already know that there's some differences from patient to patient, so to speak. That could either be genetics, it could be antigen experience of those T cells, coming from those individual donors. So there's no doubt that the quality of the T cell has an impact on it. The problem is I'm not sure that we have a really good handle at what measurements sort of define the best quality T cell. I think that's a place where we're a little bit deficient.
I just saw one email coming in, but I cannot see it.
Yep, I see it. It's basically, it says, does physical shunting of multiple receptors, for example, CD45 away from the immunologic synapse during normal T cell activation, occur during CAR activation? Second part of the question is, how are the immunological synapses different in general between normal T cell activation and CAR activation? So, I think I'll refer you to some papers that are starting to emerge that are doing some imaging, molecular imaging of CAR T cells interacting with antigen, and a number of different, TURF and other methods that are being used to kind of look at this. And there's some that are starting to be out there. The short answer is that there's no doubt that the CAR synapse is different than a TCR synapse. At least in terms of some of the arrangements. the specific question that you posed, which is sort of the exclusion of CD45 as a receptor that tends to inhibit activation at the synapse. And so, that physical exclusion from the center of the SMAC, is something seems to be important in a TCR. Does that happen, or to what extent does that happen with a CAR? And I think, I would say the answer is we really don't know that well at this point. I will tell you that, one of the things is that, I do think that that as you predict, the physical distance, so to speak, the hinge, the antigen, the nature of the antigen itself is going to have an impact on that synapse, right? With the TCR-MHC interaction, that distance is sort of fixed, right? So I think that's going to have a big difference. I'm happy to take other questions if there are any.
Give them 30 more seconds, just in case somebody type slowly.
Sure.
Well, with thanks, Terry. Personally, I learned a lot from you today. Thank you very much again.
Yeah, I'd love to say that, I have all the answers. I think, this is really one of these remarkable situations where, we now have a very effective therapy, built around chimeric antigen receptors, tremendous enthusiasm. And I think, really it's good to see now in the field, publications emerging, that really are beginning to look more carefully at the immune biology of these cells. I mean, I think we're learning surprising things, and I would predict that, in a few years, I think the structure of chimeric antigen receptors is going to look a bit different than it does currently.
It's going to be very exciting, yeah.
Yeah.
Great. Okay, I think, thank you very much again.
All right.
Have a nice day
Thank
to
you, everybody, for your attention. I appreciate those who stayed on a little longer to ask questions. All excellent questions, and I appreciate everyone's thoughts.
Thank you.
Thank you so much, Dr. Fry, and thanks to everyone for coming.
Thank you.